Other translational inhibitors that we have tried, they target a different factor, which is this EF1A. It's also involved in translation, and is also known to interact with at least one of the viral proteins. And among them, we tested ternatine-4, which has a very good IC90. Now, we are talking about 10 nanomolar, very low concentration required for making an impact on viral replication. And plitidepsin, also known as Aplidin, is another compound that also targets EYF1A and has been approved in Australia for the treatment of multiple myeloma. This one has an IC90 even better, of 1 nanomolar. It's very, very potent inhibitor, which is one of the things that you want to have when you're looking in a drug in these assays. Very potent inhibitor for a drug that has been already been used in humans, and we know that adverse events with respect to treatment, at least of multiple myeloma. We see, if you look to the cytotoxic data, the ones in the discontinued line, it looks like these compounds, they're a little bit cytotoxic because you get reduction to 50% of the cells, but it's not that they are killing 50% of the cells that have been treated. It's because during the time of the assay, the cells double if in the absence of inhibitor, and in the presence of this inhibitor, the cells don't double, they stay the same. They don't die, but they don't double, and then at the end of the experiment, if you compare them with not treated cells, there is only 50% of cells. But it's not because 50% have been killed, it's because this 50% has not doubled to 100%. So that means that perhaps we can also not have cytotoxic effects when we use them in vivo from the cells or adverse events if they're used at the right concentrations for still inhibiting viral replication. Okay, so these in vivo cells. Now we move also to human cells, to name 3Ts. And here we're comparing now remdesivir with plitidepsin or Aplidin. And what one can see is that the IC90 of plitidepsin or Aplidin in 293T cells, human cells, is sub-nanomolar, which is around 20 to 30 times more potent than the IC90 of remdesivir in the same cells for inhibiting the viral replication. So it's more potent than remdesivir. Not only works also in human cells like 293Ts, which are tissue culture cells, but they also works in primary cell, human cells. These are here in the right corner on the top. These are pneumocytes, human pneumocytes, the ones where the virus replicates, that are derived from pluripotent, human pluripotent stem cells. And in this assay, remdesivir also is able to inhibit very well at the low concentration, 10 nanomolar, the infection by SARS-CoV-2. And at the end, we did what is called a time to addition assay, which is what is seen here in the right bottom corner, in which we treat cells before infection or after infection, and we see whether the inhibitor is still giving an impact on virus infection. And Aplidin, even 4 hours post-infection, is able to reduce viral replication at the time that remdesivir is unable to, you need to treat early with remdesivir to make an impact. So it looks like in these assays, Aplidin or plitidepsin is more potent than remdesivir. But the important thing is what the protein does in vivo. Now, plitidepsin is targeting EYF1A, but there is always possibility that it's off-target activities of inhibitors, perhaps inhibiting viral replication, not by inhibiting EYF1A, but by doing something that we don't know that plitidepsin is doing. Well, it turns to be that because plitidepsin and these inhibitors of EYF1A, they have been used for the treatment of cancer, some cancers have developed resistance to this drug. And this resistance is due to a mutation in EYF1A that changes one single amino acid, A399 by V, and prevents binding of plitidepsin to EYF1A and then inhibition. Now, what we have done is we have made a cell line in which we have CRISPR-edited EYF1A to introduce this mutation, A399V. And then we look whether plitidepsin is still able or not to inhibit viral replication. That's what we see in the upper right corner. In the upper right corner, we see that in red, cells that contains this point mutation, single point mutation EYF1A, now they are unable to be inhibited by plitidepsin with respect to replication of SARS-CoV-2. Well, if we use remdesivir in the same cell, remdesivir has no problem because it has a different target. So what these experiments demonstrate is that the target for inhibition of viral replication of plitidepsin is indeed EYF1A. Remdesivir is being used, so probably for new inhibitors, they will be using in combination with remdesivir. So it's always good to see whether new inhibitors they are synergizing or they have additive effects, or perhaps antagonizing, so they don't work, they kill each other with remdesivir. In the case of Aplidin or plitidepsin, we did experiments in which we combined remdesivir with plitidepsin or Aplidin, which they are here summarized. And in this case, what we have is an additive effect. That means that one drug, the two drugs can be used, and not only that, because it's an additive effect, probably you need a little bit less of each drug in order to make the same impact. Now, does it work in vivo? For that, we have done some mouse studies. Mice are not being infected, at least with the SARS-CoV-2 that they were being circulating before variants came, and that's the experiment that we have done here is before the variants. And for that, we need to make, we can make mice able to replicate the virus, but we need to do tricks. And the tricks is make them to express the human ACE2, because the mouse ACE2 is not very good to allow replication of SARS-CoV-2 in the mouse. You can achieve that by transducing mice in the lung with an adenovirus vector that expresses human ACE2, and 5 days later, after this transduction, now the lung cells of the mouse are expressing human ACE2, so you can challenge them with SARS-CoV-2. And when you do that, now SARS-CoV-2 will replicate, and replicates, as we can see here in the right, to levels around between 10 to the 5, 10 to the 3 PFU per ml at day 3 post-infection. Now we are taking these mice and treated with Aplidin or plitidepsin or remdesivir, different dose, 1 mg per kg, one single dose, 0.3 mg per kg, 3 doses, and compared with remdesivir, 50 mg per kg, 3 doses. And what we see is that both remdesivir, but importantly plitidepsin, at as low as a smaller concentration, is able to reduce viral replication to a level that in some instances we cannot even detect viruses in the lungs of these animals. The other system that we have used is not a transduced mice, but a transgenic mice. In this case, these are transgenic mice that they are available from Jackson Laboratories that express human ACE2 under the control of a keratinocyte promoter, K18. And in this case now, because this results in better expression in a lot of cells of human ACE2, the replication of SARS-CoV-2 in these animals is much, much higher. It goes to 10 to the 7 PFU per ml. And when we did the same treatment here, 0.3 mg per kg 3 times, we again were able to reduce dramatically the replication of the virus, indicating that, yes, Aplidin or plitidepsin is making an impact on viral replication in the lungs of this animal. Does it also reduce disease? For that, we look to the lungs of these animals, and we see that in untreated animals, there is a lot of inflammation, both in the alveoli and in the bronchia. But when we treat the animals with plitidepsin, we see very little inflammation. It has been reduced a lot, meaning that there is a big reduction of disease. Remdesivir reduces inflammation, but not as good as plitidepsin. And in uninfected mice, obviously, there is no inflammation. Now, what happens in human? Plitidepsin has been developed by a company called PharmaMar, again for the treatment of myeloma, but they are very interested also in seeing whether plitidepsin is able to be used for the treatment of COVID-19. They have conducted already phase 1-2 trials that shows no severe adverse events and potentially some impact on viral replication in COVID-19 patients. And now they are going to start basically clinical trials. They are already in clinicaltrials.gov being described, which they will involve a total of 600 participants that have been randomized into 3 groups, with one group with 1 dose of plitidepsin, the other group with a different dose of plitidepsin, and a controlled placebo. All of them will receive obviously standard of care. This remdesivir, and the idea here is to look whether now treatment with plitidepsin results in less days of hospitalization of people with COVID-19. So in summary, with respect to antivirals, I show you how you can use information about virus host protein interactions in interactome. We have also used the phosphoproteome. I didn't present it here data, but we have some inhibitors also based on proteins that have been phosphorylated. And that provides a rational therapeutic target for drug repurposing, and how plitidepsin, which is an inhibitor of EYF1A, has potent SARS-CoV-2 antiviral activity both in vitro and in vivo, and phase 3 clinical trials are moving forward. So hopefully they will make also an impact for the treatment of COVID-19 in patients.